This is a lecture for my Bio 410, Bio 510 Bioinformatics class on the topic of transcriptomics. And today I'm going to be talking about transcriptomics broadly to provide some background and understanding of the different approaches for investigating transcriptomics, understanding some of the means that we use for assessing transcriptomes, and also digging a little deeply into the how we are actually understanding and interpreting gene expression data. So I think when we think about transcriptomics, this is a helpful diagram that I found on the web, which indicates that when we think about molecular biology, there's sort of some key terms and ways that we might think about understanding molecular biology, right? And in this case, we have our central dogma approach of we have genes which are then transcribed into proteins. Those proteins are then folded. We end up with three-dimensional structures, and then based upon their forms, they're going to provide certain function. And in turn, often those functional components will be regulating which genes are actually going to be expressed. Well, when we're looking at transcriptomics, we're specifically focusing in on what are the patterns associated with the RNA that are being transcribed. And I think it's important for us to remember here that, when we look at transcriptomes, we are looking at the nature of different patterns of gene expression, right? So in every cell in an organism, basically it's going to have the same genome. But when we look at different types of cells, whether they be neurons or cardiac muscle or kidney cells, they're going to be expressing different genes from that genome. And the patterns of gene expression that they exhibit will ultimately contribute to the function of those cells. So in understanding the patterns of gene expression, we would hope to get some understanding of their underlying phenotype associated with these patterns of gene expression. When we talk about transcriptomes, basically what we're talking about is the identity and quantity of all the RNA that's expressed from the genome in a cell or a sample of cells. Additionally, we feel like there's a relationship between this transcriptome to something we would refer to as the proteome. And the proteome would be the identity and quantity of all the proteins expressed from an entire genome in a cell or a sample of cells. And as identified in the previous slide, unlike a genome, when we look at these patterns of transcriptomes and proteomes, they can really vary from one cell to another depending upon what is the functional phenotype of that cell. When we're looking at some of the basic principles of transcriptomics, we also want to understand that genes are regulated, right? They're turned on and they're turned off at different times. And of course, this goes back to some of the original work by Jacob and Monad on bacteria, in which they discovered operon structures, where they showed that there was this pattern of growth in bacteria where they would initially grow on glucose, but once all the glucose was consumed, they would sort of lag for a little bit, and then they would start to be able to consume lactose. And they found that this delayed switch was due to transcriptional upregulation of lactose metabolism genes. And so what we're seeing here is a big change, right? It's the same organism. At one point it's metabolizing glucose, and then it changes the nature of the organism so that it will now consume a different energy source, right? So some genes are being turned off and other genes are being turned on. And so through a wide range of different types of experience, both mutational experiments and enzyme kinetic analyses, they worked out that there was a whole regulatory system that was basically turning these things on and turning these things off. And some of the key components involved in this process were regulatory genes and promoters, right? And these different components were able to turn on a whole set of genes that belongs to an operon. In this case, what was referred to as the lac operon. So it's important that understanding that there were structures that were actually regulating the expression of these genes, and how these genes, these regulators were being interacted with, was actually determining what the patterns of gene expression were. So when we think about the activation of genes, we're often looking at sort of two key components, or some key components that are typical in this case, are response elements. And so these are segments of DNA that bind to protein activators and repressors. And when these DNAs bind with this protein complex, those patterns of binding of these different complexes will actually have a lot of influence on transcriptional rates. And a classic one is the binding of transcription factors to then recruit RNA polymerase II. Enhancers are another important component in this equation, and these are often regions of the genome that are a little bit away from promoters. And depending upon their location and orientation, they can have a specific effect on what patterns of gene expressions are as well. So here is a classic example of how an enhancer and a promoter might work together. So here we have different transcription factors that bind to these different regions. And when we get the binding of transcription factors to both the enhancer and the promoter, we end up creating they end up binding to each other, creating this loop. And this process of having this protein complex form then facilitates the recruitment of RNA polymerase II to this coding region, and we thus can get RNA synthesis or transcription. Now, there are a lot of ways that people have looked at transcription over the years, and I'm going to talk about a few of them here to kind of bring us up to what are some of the tools we're using in modern transcriptomics, so then we can start to talk about some of the tools that we use to look at them. So when it comes to transcriptomics, early on there were techniques such as nuclear run-out experiments and northern blotting experiments. And these were experiments where basically we were using some sort of labeled nucleotide, typically a radio-labeled nucleotide, to estimate what were, the amount of transcripts that are being produced in a nuclear run-out experiment, or using radio elements to sort of tag specific RNA species so that we could then observe them in a gel or a blot. While there's not a whole lot of nuclear run-out experiments done these days, so they're not as commonly used. They're using some sort of specific techniques. But northern blots are still sometimes implemented, and basically the process was you would take RNA, separate it on an agarose gel, transfer it to a membrane, and then you would use a radio-labeled probe to identify different things on that membrane. And by using that radio-labeled probe, you could identify where an RNA species that probe would bind to would be on that membrane, and thus sort of like where it would have been on that gel, right? And so on the right here, you can see an example where we see an alternative transcript, something that was much smaller than what was typically expressed in other genes. So in this case, whatever transcript we're looking here in brain and placenta and lung and kidney was expressed a transcript that was approximately 4.4 kilobases long, whereas the transcript that was expressed in heart was 1.4 kilobases long. So this shows that we have an alternatively spliced transcript active in this case. These days, I think the place where I see northerns used more frequently than in other cases would be in the identification of things like small RNAs and non-coding RNAs in certain samples. After some time, the molecular biologists figured out there was an easier way perhaps to deal with RNA transcripts, and that was to instead of trying to deal with RNA, was to create, which is incredibly volatile and hard to deal with. It breaks very easy. It breaks down very easy. It's really difficult to deal with, is to stabilize it when we're in the lab by creating something known as cDNA. And cDNA is, the C stands for complementary DNA. And so this is a way to make sort of double-stranded DNA from single-stranded RNA. And there's a small diagram here over on the right that explains this process, wherein you basically have some sort of primers or adapters that are used to generate a first strand match to our RNA sequence, to our RNA species, to our RNA transcript. And then once you've got that first strand match, you'll then create a second strand to it. And then typically there'll be a step once you've created cDNA that you might do something like PCR amplification to make sure you have increase the copy number of the things that you're looking at. And this actually has become the basis for a lot of really important molecular techniques that we still use today. One of the most common applications of using cDNA is in the process of quantitative PCR. And this was a method that was developed to actually calculate the quantities of different transcripts and samples. Basically how it worked is in the same way that PCR would target a specific target in the genome, quantitative PCR would target a specific cDNA transcript. And what it would do is, as you would sort of run PCR on that sample, all of the primers would have a fluorescent tag attached to it, and that fluorescent tag would then be observed with a camera, and when you hit a certain threshold, it would record the cycle of PCR where it hit that threshold. And then that would be used to identify approximately how much, what sample of a specific target you had in your sample. And there's a relationship, inverse relationship between CT cycle, or the number of cycles of PCR, and the concentration of whatever target you were looking at. And so as the cycles go up, you actually get, it would be measuring a lower and lower amount of transcript in your sample. One of the interesting things about qPCR is, while you may measure the CT cycles, the CT cycle in itself needs to be pegged to something in order to make it a meaningful value. And so you either have to make often you would make something like a standard curve to associate it with, or you'd actually, the more common approach is to say, what is the CT cycle relative to a known housekeeping gene? So a gene which is consistently expressed across a lot of tissues. So you're looking for differences in gene expression. And so how you identify gene expression is by looking for differences in the relative expression. So the expression of something relative to a housekeeping gene. And so, GAPDH, actin is another common housekeeping gene. And basically what you would do is you would look at the fold change of gene expression between two samples by making what you would measure to be pegged to a specific housekeeping gene. So so GAPDH presumably would be expressed equally across multiple tissues. And so then you could look at two different samples and you could see whether or not expression was the same. So the differences were the same, pegged to this GAPDH value, which should be consistent across all samples. There are some other normalization assumptions that you have to make in this process, but it's a pretty accurate way, perhaps what we would consider to be the gold standard for estimating RNA concentrations if done well. After qPCR in the 90s was the advent of the microarray. And what the microarray did is it took cDNA and it actually labeled it with some specific markers. And then it took a whole bunch of probes and it washed those markers over it washed these cDNA that were labeled across a chip, which would then hybridize wherever these cDNAs would match this chip, and on the chip were all these probes. And wherever it matched and bound to the surface, it would indicate that sample was probe, the thing that matched the probe, whatever gene it was actually in your sample. And so basically how it worked is first you would harvest some cells, you'd purify some RNA, you would then reverse transcribe that RNA depending upon, you usually have two cases, and you reverse transcribe it either with a Cy3 or a Cy5 labeled cDNA. And so you'd end up basically with green and red samples. And then you would take this cDNA and you would flow it across a chip, and it would where these cDNAs would find a match to these probes, these sequences would match these probes, you would get the green or the red binding to the chip. And then you shoot it with a laser, and depending upon the color that was emitted, you would know whether or not that probe had found its match, right? And every one of those probes, those oligo probes, those oligonucleotide probes, was associated with a specific gene. And then what you would do is you plot the intensities of these lives, and then you'd be able to see sort of, what was the relative gene expression based upon how brightly green or how brightly red these samples were. And a classic way of observing this thing was to look at a heat map, right? And so where we see red versus green, we could see that genes were, going from one, where they were expressed in one stage, they would see a different pattern of expression in a different stage. And so we could, through these experiments, we could see, very different processes, like as we would go through sort of wound healing, that different genes would be turned on and different genes would be turned off based upon, what was going on in development. So this was one of the ways that we could sort of start to understand how transcription was happening to a lot of different genes all at one time. qPCR was targeting just a handful. Microarrays allowed us to target sort of thousands of genes in different cases. Then that led us to RNA-seq. And RNA-seq, basically RNA-seq is an abbreviation for RNA sequencing. And one of the real powers of RNA-seq is, while microarrays depended on only looking at the things that we actually had sequences for, we had to already have this chip with these oligonucleotide probes that were attached to it. So we could only look at genes that we already knew something about. Well, for RNA sequencing, we actually don't need to have prior knowledge of what we expect to see in our sample, because we're going to sequence everything. And so it's actually possible to discover new things and new transcripts actually while we're doing these experiments. And one of the other really powerful things about it is that this process is quantitative. So as we do sequencing, the amount we sequence of one gene is actually representative of how many transcripts for that gene are actually in that sample. So there's a quantitative component, which is really powerful for us. And when we do RNA-seq, basically what a typical process will entail is we'll take a piece, samples, either of cells or of tissue, we'll extract RNA from it, and in that total RNA, the vast majority of that is actually going to not be mRNA. It's going to be stuff we're going to be a lot less interested in. It'll either be ribosomal RNA or tRNAs, right? And those are going to basically all look the same. So it's not very interesting to us because that's not where we see the variation among samples. However, there's a bunch of different methods that we can use. Either we can make use of the fact, if we're looking at eukaryotes, that all eukaryotes have generally will have these poly A tails associated with mRNA, which allows the mRNA to help get transported out of the nucleus and targeted towards the ribosomes. But in this case, what we can actually use, we can use probes to pull down all the things that have poly A tails on them to isolate the mRNA away from the ribosomal RNAs and tRNAs and other RNAs we don't want to sequence. And what this does is it makes the process of RNA-seq a lot more efficient. There's also a method by which you could, instead of just pulling down everything that has a poly A tail, you could just pull out all of the ribosomal RNAs and tRNAs, and that's a process that we refer to as ribodepletion. And one of the powerful things about that process is it leaves behind maybe some of the non-coding RNAs that might be expressed, and that also can be important in gene regulation. Once we have our isolated and our RNA sample, though, what we can then do is we'll fragment that RNA. We'll then undergo that cDNA synthesis step, where it will generate cDNA to sort of stabilize our sample, put some adapters on the end, and then we can go ahead and do the use a next generation sequencing tool like Illumina sequencing to then generate the sequences of the RNA that we need. Once we have those sequences, we're looking at pieces of RNA that are relatively small. There are some different ways that we can take that. A classic way, if you have a genome, is that you'll take these reads, and we talked about this a little bit before, we'll map these reads to an actual genome. Once we can do that, we can generate assembled transcripts for all of the alternatively spliced transcripts from that genome, and actually end up with the sequences that we'd see. And you can see this is a track of what that mapping might look like and how it translates into different alternatively spliced transcripts. And one of the key things here is that we have reads that are kind of broken in pieces, right, where they map across two different exons, right? We see some sort of exon to a read that maps to one exon, another exon with an intron in the middle. And this helps us identify these patterns, these gaps that we see here. Now, that's not the only way that RNA sequencing works. So in addition to doing sort of that reference-based approach that we saw, one of the powerful things about RNA sequencing is that when we sequence it, we're sequencing the entire genome, and this lets us look at things like alternative splicing, which when we looked at microarrays, we couldn't have done before. But if we aren't actually, it allows us to actually look for some things that we might not see, and so it actually lets us find transcripts that we might not have anticipated being our sample because we're just mapping it to the genome. But there is also like this inductive and deductive component to it, because one of the things that that you can do when you do RNA-seq is that you can actually, if you do de novo assembly too, along with some of these mapping methods, you can discover new transcripts that you hadn't ever hoped to find. But you can also set up very specific things where you might not, you want to do some specific tests to look and sort of say, well, what are the patterns of gene expression we see between very specific samples? Example of how that might turn out is over here on the right. We have some data that I generated for a study that I did where we were looking for gene expression patterns associated with fish that have placentas versus fish that don't have placentas. And what we found, you can see here, is that there's a whole bunch of expression in this transcript right here. And this transcript has something known as a TLV coat protein. This is an endogenous retroviral coat protein. And what we see here is that we had a whole bunch of expression of this transcript in one of these organisms, but not in this other one. And this one is a less placental, is a lot less capacity for giving post-maternal provisioning. Now, we expected there to be differences between these two organisms. So in that case, we're kind of taking a deductive approach where we're doing a comparison between the two, but we didn't know which genes they would be. And so we were able to discover a significant difference between these groups, something that we didn't anticipate finding from originally in our sequence, as we designed this experiment. Another thing that I'll point out is that for these organisms, we actually didn't have a genome for them either. And this is also one of the powerful things about RNA sequencing, is that we can actually generate transcriptome sequences for these organisms without having to generate the genome. And sometimes it can be very challenging to generate a genome sequence for an organism, and it's a lot easier to generate RNA-seq data, and the amount of RNA-seq data that you would need just to create a transcriptome. So there's a lot of power in being able to take this approach. Again, and one of the other powerful things about this is that there's a lot of, it's quantitative as well as in this process. So all the reads that we're sequencing from this are actually going to be, we can use as a proxy for level of gene expression. The dynamic range, as we talked about in the papers on dosage compensation, is much better in RNA-seq than for microarrays, and we have less detection bias as well. There's also some really powerful things here too, where we can do some strand-specific analyses, where we can actually identify, which strand the sequencing is coming from. So that can tell us a lot about whether or not we think what we're looking at is a gene on the positive strand or the negative strand. We know that sometimes genes can overlap, but also it could help us see if there are any sort of regulatory things going on where we're actually seeing negative expression that might help inhibit gene expression. And the other really cool thing is we can do some really small amounts of RNA. Single cell RNA sequencing is a very up and coming field, along with spatial transcriptomics, where we're putting RNA-seq data in the context of its location within a tissue. And single cell data is a way for us to really get a level of detail that we haven't been able to have previously. And I mean, we're just looking at the RNA from just one single cell. I mean, that's kind of incredible. We can generate good data from that. I think it's important when we think of it. We talked a little bit about this already in the mapping section, but I think it's important for us to think about, how we are actually calculating and examining this gene expression data. And just like with qPCR, a lot of RNA-seq analysis, what we're doing is we have to find some way to normalize our data to sort of, and a lot of these analyses are actually, we're looking at the level of expression relative to something else. So in the qPCR case, we're looking at it relative to a housekeeping gene. With RNA-seq data, that may not be as necessary that we actually have a housekeeping gene, but what we are looking at is we're looking at expression in one sample versus another sample. And over the years, there's been a wide range of ways that we would estimate gene expression. And I talked a little bit about this before. There are estimates like RPKM and FPKM and TPM. And what all of these are is they're efforts to normalize our data. And we're trying to normalize the data to remove some of the systemic variation that's going to happen in our data just to the nature of RNA-seq and the nature of the analysis. And by doing this, we're going to allow for comparisons to be made across studies. And there's a lot of various ways to accomplish this, but they all attempt to do the same thing. But each one has their own specific idiosyncrasies. And so it's important that we think about why we might pick one analysis type over another. And so if we just look at those three metrics, RPKM and FPKM and TPM, we can see that RPKM, these are really simple ways of doing normalization, right? We talked about this earlier. So, we count up the total number of reads in our sample, and, usually we're going to have so many millions of reads, and that's going to be our overall scaling factor. And then what happens is we'll look at a specific target, and then we'll divide the reads that we found at that target by our scaling factor, and then we'll divide it by the length of whatever that target was. And that's because if I have a transcript that is 10 times larger than another transcript, it should create a lot more reads just because we fragment them and break them up, right? So it's going to have more sequences that map to that target just based on the fact that it's longer than another target, right? It's got more sequence in it. So we have to control for that factor. FPKM is basically the same as RPKM, but FPKM is just made for paired end reads. It's these fragments. So here we have to account for the fact that when we're counting reads, we actually have 2 reads associated with each fragment. TPM is similar to both these, but it just kind of changes the order of the process by which this happens, right? So instead of dividing by the scaling, the library sequencing depth scaling factor first you actually divide it by the transcript length, and then you divide it by that scaling factor. And that's what's going to give you TPM. So these are basically described as like reads per kilobase per million, fragments per kilobase per million, and transcripts per million. And one of the reasons why you might think about this is they're all relative. In these cases, these are all relative values. And what they're actually looking to do is compare the relative proportion of reads that have been mapped from samples. And one thing I think that's key to note here is that there is some benefit over perhaps taking one approach over another. So if we were going to look at RPKM, right, I think one of the important things here is that when you use TPM versus RPKM and FPKM, TPM is going to you're always going to be able to sum up all the TPM values in a sample. It's always going to be the same. So it's easier to compare proportions because they're all basically going to sum to the same number. Whereas we look at RPKM, we will see that the amount of RPKM will change based upon these things. So if we're going to look at multiple replicates of these, right, we can see that if we were to sum up these values, we're going to end up with different actual totals, right? So the sums of each of these columns can be very different, right? Whereas we notice if we do TPM based upon the same process, we actually end up with the same values. So when we look at the proportions and the change among these different samples, it becomes a lot easier to compare because we're talking about a proportion of a total of 10 in each of these cases. Where in this case, we're comparing a proportion of a total which is constantly changing. So that's why I would generally say TPM is a little bit better for comparing these relative changes among samples. Ultimately, though, when we're looking at transcriptomics, what we're often really interested in, just like we were when we looked at gene expression data using qPCR and going back even farther, we're interested in identifying what are differential patterns in gene expression. We go all the way back to the work of Jacob and Monad, right? And in that case, we're looking at how we get this switch from glucose to lactose metabolism, right? What we want to figure out is what are the genes that are driving that pattern. And basically, a common way of doing this is what is essentially a t-test, right? So if we remember how t-tests work, right, we're going to have one distribution of values, and we're going to compare it to another distribution of values. And when we're looking at significant differences, we're looking to see if these two distributions are actually overlapping with each other. Now, when we do RNA, and basically, what is significance is when there's not a whole lot of overlap, and when when there's not significance is when there's a lot of overlap between these underlying distributions. Now, transcriptomics is way more complicated than doing just simple t-tests, but the base premise for it is pretty much the same thing, right? We're going to be using some sort of advanced statistical model to identify these differences in gene expression, right? The one condition versus a different condition. And so there's a whole bunch of different methods, and this is just a list of some of them. There's a lot of them. And I'm going to talk a little bit about DSeq2. And so DSeq2 is a little bit different from the FPKM, RPKM, TPKM methods I talked about before, where it does do some read normalization, as we talked about, but it does it using a slightly different methodology. And I'm not going to go into a lot of detail about it, But basically, it's going to use counts divided by some sort of sample-specific sizing factor. And that sizing factor is determined by looking at the median ratio of gene counts relative to the geometric mean for each gene. And so it's going to calculate these sizing factors. And then what's it going to do is it's going to use those size. It's going to, after it has normalized your data, it's then going to take those counts and it's going to estimate something called a gene-wise dispersion. It's going to estimate these gene-wise dispersions. And what are gene-wise dispersions? Well, they are the variances in gene expression for given values of gene expression. And one of the things that you notice, if this is a plot of the sort of means and variances in expression in counts, right? And so as you increase the mean, notice that the variance becomes less between things that have the same means, right? So like the variance in expression is less, in these counts is less as we go up. And so at low values, we see that gene counts are a lot more variable. So counts of genes are a lot more variable. And so because of this, like this isn't just, we can't assume that sort of variation is consistent across all samples. And we have to account for the fact that when we're looking for differences, what's a significant difference, we might need to treat it differently when we're talking about things that are lowly expressed versus things that are highly expressed. So one of the things we do when we do that is we actually, there's a way that it goes about sharing information to calculate these dispersion values, these estimates of variation, because they're unreliable when there's not a lot, when there's low sample size. They actually will share the results for things that are all sort of in the same level of gene expression. And by doing this, right, they'll basically be able to shrink these dispersal levels. And what happens is they build a rather complex model, right, where they have these normalized counts that they've generated using these size factors. They'll have their estimates of dispersion or variation, and based upon this process, what they'll find is this kind of pattern of gene expression. And what the really complicated part about this whole process is you'll note that here that there's these blue lines up top and then there are all these blue lines here. There's kind of two different models at play at this. There's a bunch of models that are going to actually shrink your data. They're going to actually use these dispersion estimates are going to be shrunk towards this model because this model is seen as being the overall pattern. However, there are some things that have extremely high values in dispersion above the model. And these are your highly expressed genes. And due to biological and technical reasons, they actually behave differently for real reasons than the rest of the data. So it actually goes through and it identifies sort of these two different categories of processes between sort of large effect and weak effect genes. But once it shrinks these gene-wise dispersion estimates, then it'll actually test for significant differences. And one of the things that it's doing is when it does this shrinking, what it actually does is it has an effect when we're thinking about gene expression data. Often we're looking for differences that are based on what are known as log fold changes. So it's looking at, what's the fold change? So is it twice as highly expressed? It's a log fold change. It's a log 2 change, right? It's looking at these log fold changes. And if you don't do that sort of shrinkage process, you end up with a process in which we have a lot of noise, a lot more noise in our data set. Whereas if we're able to do that shrinkage, it allows us to have a much clearer interpretation of what's going on in our data set. And so what we're then allowed to do is compare between two samples to see which examples we see sort of overexpressed in one tissue versus another, or underexpressed in one tissue versus another. And so these are examples of how these plots, this is what's known as an MA plot, we can see the difference in gene expressions, right? And to do that, we actually use something known as a negative binomial generalized linear model. And this is basically a specific kind of generalized linear model, which we can use for significant testing, where we know that the variance, these overdispersion estimates, actually exceed the values of the mean. And it's a very useful model in this case. And that's actually how we're testing for the differences between them. So there's all these very specific criteria that are associated with this algorithm that are really important. And you can see that it's a lot more complex than your average t-test, right? Like, and the reason I went into detail of this, one of these, is I wanted you to get an appreciation for that. There's a lot of advanced methods that go into this. Now, conceptually, in terms of what's going on, I think you can understand that it's equivalent to this other process. But because it doesn't behave, this data doesn't behave like other kinds of data, we have to have these more complex models to generate what is effectively our null hypothesis. Now, there's a whole other step too that we have to take into account, is the fact that when we're doing these comparisons, we're not just looking at like one case in a t-test. We're actually looking at thousands, perhaps tens of thousands, or even more significant tests. And based upon just random chance alone, it's very possible that we could see significant differences just due to random chance, right? So if I test something so many times, I would expect to see, 5. If I have a p-value of 0.05, right, I would expect to see 5% of my tests to be significant just by random chance alone. So to control for this, what we call multiple comparisons problem, you'd use an approach that controls for something like the false rate of discovery. So historically, for multiple comparisons, we might use something like a Bonferroni correction. But in this case, because we're doing so many tests, it really is overly conservative. So using a tool like a false rate of discovery correction is one of the ways that we control for this issue of multiple comparisons. Another key part of looking at transcriptomics data is by looking at clustering. And so clustering is a way that we can identify genes that are co-expressed. We can look for similarities between samples or similarities between genes. And this is really sort of a pattern or inductive way of looking at our data. It could be deductive if we came up with a hypothesis ahead of time, but a lot of times this is a very inductive process. And I'm not going to go into a lot of different clustering methods, but I am going to talk about one because hierarchical clustering is kind of like the basic way of approaching clustering, and it's instructive for letting us understand how a clustering algorithm works, and there are other ones that work slightly differently, but they work in somewhat similar fashion, I think. So when we think about how hierarchical clustering works, what it does is it calculates the distance between all genes, and then what it finds is the smallest distance. So in this case, the smallest distance it finds is the distance between G1 and G6. And then what it's going to do is it's going to look for these patterns of similarities, and it's going to fuse these small clusters together. And after it finds a small distance and it fuses the cluster together, then it's going to repeat the whole process over again. And it's going to repeat again until it draws this tree from this process. So it's going to start off and it's going to find these differences, and then it's going to look for the next difference that it can put together. Then it'll look for the next difference that it can put together. And it's basically just drawing circles around each, or ovals around each of these groups until it can have all the groups into the same tree structure. And so, a way that we could think about is G1 is most like G8, right? So it's going to draw this line. Then G4 is most like G1 and G8. So then it's going to bring it into this cluster. And then it's going to keep on going. G5 is most like G7. So that's the next one here. So it's going to draw a cluster there. Then it shows that these two clusters are most similar to these two this cluster over here. And eventually it's going to do that part until it has identified what are the groupings of all the things that are more similar to each other than they are to other things. And intuitively, I think this is an easy way to think about how we might understand samples and their relationships to each other. So what else can we use transcriptomics data for? Well, there's lots of things we could look at when it comes to networks, and specifically really complex networks. And you can create things like this, often referred to as a hairball plot, right? Like this thing is pretty messy. But what you can see is, if you were looking at it, is you can start to identify what might be networks or groups of genes that might go through some sort of node or control or hub of gene expression. And so in this process, we can see that there are certain nodes that have a lot of connections, that have a lot of edges attached to them, right? And they may be really important for these networks. Also, we can look at all the genes that are under control of this and see that their patterns of gene expression might all be dependent upon some very key signalers. So this is one of the ways that we can help understand how networks are starting to work in biology. There are some interesting methods that apply this. One of them is something known as weighted correlation network analyses, or WGCNA. And basically, this is a way to find clusters of highly correlated genes where you don't know if they exist or not. And it can be really powerful, because what you can do when you do this process is you can start to identify networks of co-expressed genes that you can then look at and identify where these genes de novo are working. So you might find interesting drivers of gene expression or members of these co-expressed groups that you didn't even know about before. It also helps you to identify what are some of these hub genes that might be driving these groups or these eigengene groups. I think another really interesting application of transcriptomics data is in the field of genome scale metabolic models. And so these are models where they're actually trying to model all the biochemical reactions that are going on in organisms. And one of the ways that you can actually model what's going on in these processes is by looking at RNA-seq data, because it'll tell you which biochemical processes are turned on in a sample and which ones are not. So there are a bunch of different ways. These models have evolved through time, but basically what these models are, and they all come more from an engineering perspective than maybe from a biological perspective. But what these models are is they're models of all the biochemical processes that could be going on within an organism. So this is an example of different types of models that have been created for the human system. We're actually now on Recon 3D. We're no longer on Recon 2. But these are how we can see, like, these are the number of reactions that are occurring in these different groups. And you're actually modeling, when you look at one of these genome scale models in humans, you're looking at something like, well, this is just for Recon 1 and Recon 2. For Recon 2, you're modeling about 7,500 different reactions with all the different metabolites that are included, right? So a lot of these reactions are connected to each other. Remember, I showed you some examples of what a keg diagram looked like in class the other day, right? They're like these interconnected network and circuitry processes, right? And one of the things about genome scale metabolic reconstructions that is really cool is that they have this sort of known structure, right? And it's tied to things like metabolic processes, which are very closely associated with what we might consider to be the phenotypes of different cells. Now, I will say that they're rather hard to generate, and they have maybe certain metabolic processes are not as well identified using RNA-seq data, or even well annotated. But you can think of it as being a very powerful model. And basically how it works is we have a set of reactions, and what we end up doing is creating what's known as a stoichiometric model, where we'll have a series of reactions sort of like in each row and the metabolites in each column. Based upon these relationships, we'll be able to create patterns of biochemical metabolites that are all related to each other. There are some advanced methodologies that we use to do this. And once you create this sort of this stoichiometric model with all of these different biochemical processes in it, one of the cool things is that we can have these, it puts constraints on your system. So we're no longer thinking about that we can just move anywhere in our state space. We're actually restricted by that model, right? And you can do experiments, where you actually say like, hey, I'm going to add in a whole bunch of carbon dioxide and I can stress test these systems and see what's going on in these metabolic processes and make predictions about what you expect to happen based upon real data, RNA-seq data. And so, again, this is what one of these network models looks like, right? Where we are we have these different metabolites. They are creating, we have different sort of reactions that are creating these different metabolites. And based off reaction one, right, we're gonna be consuming something and producing a 3 and a 4, and so we can start to fill out this matrix, and by filling out this matrix we then can actually model the dynamics of the system. And so an example of how we might use data like this is, instead of just looking at RNA-seq data, this is some work I did with a student quite a few years ago. Blake is now a PhD student at Brown. What we did is we used single-cell RNA-seq data to make genome-scale metabolic models for all of these single cells. And then we tried to figure out, can we actually cluster, say, for instance, different breast cancer cell types by their metabolism instead of their RNA-seq? Does that give us a different way of thinking about these samples and what might be driving them? And it's another way to think about how we can use this data in another fashion. We're not just looking at RNA-seq is definitely a phenotype. We're looking at a phenotype which is basically based off of metabolism. And for something like cancer, where we could imagine that they could have different types of cells that are doing different metabolic properties, it can be really interesting for us to think about how we might have cells that have sort of one metabolism if the ER positive and doing a different metabolic processes as well. So very interesting to think about how we might look at these cells in different ways based upon a different type of phenotypic measure. And again, this is all from RNA-seq data. So it's just another way that you can use RNA-seq data. Now, we're not just looking at transcripts now, we're looking at it at a different level. All right, so these are some of the ways that we can look at transcriptomics data, and we'll hopefully get a little bit of a chance to practice some of these things in class with some data. All right, thanks for your time.